My name is Eric Williams and I'm a postdoctoral fellow in the laboratory of Lenny Guarente. I study this class of proteins called sirtuins. If you activate sirtuins in lower organisms like yeast, worms, and flies, they'll live longer. And they seem to be beneficial for a lot of aging-related diseases in mammals, like diabetes, neurodegeneration, and cancer. We had the hypothesis that they might be involved in stem cell regulation. So for example, in your skin, you'd have some stem cells that are deep in the skin, and they are dividing and forming all the cells that are on the top of your skin. And the cells on the top of your skin are differentiated and they are protecting you from environmental factors, but they are continually dying. So you need a completely new set of these cells to have a functioning skin. If the stem cells are no longer able to divide and recreate the skin, then a process of aging is occurring, which could also be the same process that's causing a lot of diseases. So in this experiment, I took some embryonic stem cells. These are cells that are able to turn into any cell type within an adult organism. And I made some red ones, which are wild type, they have SIRT1, and then I made some green ones, which have everything except for SIRT1. And then I injected them both into a 30-cell developing embryo, which then developed into an adult mouse. In this experiment, you can see the olfactory system of the mouse, so that's the nose of the mouse. And you can see on the left side and the right side there are these neurons that are red and green, and it turns out that they're mostly green. So in this case, those cells that didn't have SIRT1, the ones that are green, seem to have a preference for making these olfactory neurons. This might hint at some kind of molecular switch in which olfactory neurons need to turn off SIRT1 in order to make these types of neurons. Mice are actually amazing smellers. They have approximately 1,200 different types of olfactory neurons. So as a comparison, humans only have between 100 and 150 types of olfactory neurons, and dogs, which we think of as one of the best smellers in the world, only have around 400. So what you're looking at in the middle of the picture, where all the green signal is, those are the neurons. You can see these little spaces in between, and that is actually where the odors come in through the nose, and they activate those neurons, and then those neurons then project to the higher order centers of the brain.